Evolution. We are discussing biological evolution. Formation of protobionts. Formation of what you call protobionts. Protobionts. First, what happened? Abiotically. What happened? Abiotically, molecules produced. These abiotically produced molecules self-assemble. Okay, self-assemble and into droplets. Okay, then encloses watery solution. Okay, and maintain chemical environment. What they done? Maintain chemical environment. Okay, and this environment, different environment from surroundings. Okay, then scientists, all these peers. So this one is called, scientist is called, what he called protobionts. What scientists called, these all are protobionts. Next, this is the one thing, the liposomes. Okay, the liposomes, okay, are lipids. Okay, in the solution that can very very important, self-assemble and form lipid bilayer. Okay, next, some of the proteins. Okay, some of the proteins inside the liposome. These proteins, okay, acted as enzymes. Properties of enzymes. These enzymes, fast multiplication. Because of these enzymes, fast multiplication happened. Fast multiplication of molecules happened. This all we discussed. Again we are recollecting. Then next concept, what? This is the most important. The coacervates. What do you call coacervates? The coacervates. Okay, this is important. The coacervates with nucleoprotein. Coacervatives with nucleoproteins. Okay, and nutrients. Okay, nutrients. Add limiting surface membrane. Very very important. Limiting surface membrane. This all, what happened? This all togetherly forms virus. Okay, so or also we can call free living genes. These points you should have to remember. So these all genes united to form protoviruses. So in present day later they converted into or similar to present day virus. These points you should have to remember. So this is the one category. So what we discussed, observe carefully. Formation of our content is formation. Already we discussed this topic in yesterday, but again recollecting the information. The formation of protobionts, how this happened. Abiotically produced all molecules spontaneously. Okay, spontaneously spontaneously assembled and converted like a droplets. These droplets enclosed by which one? The water solution, and this environment completely, this chemical environment, this environment is different. So this will be considered as which one? Protobionts. What you call? Protobionts. So liposomes are there, no lipids. What you call? The lipids which are there, they will be solution self-assemble. These all self-assemble. These all are lipids. These all are self-assemble and convert into what? Another layer. What you call? This one is called lipid bilayer. Then this will be converted as a what you called like protobionts. So the protein, these are the proteins which are present. Next one, the proteins which are present in this liposome. The proteins which are present in the liposomes acts as enzymes, and because of these enzymes, what happens? Self multiplied, multiplication of molecules happened. So this is the what we call protobiont. This is the total structure of protobiont. So that only mentioned droplets, enclosed watery solution, okay, and chemical environment is different, then forms a protobiont. The liposomes are the lipids, solution lipids, self-assemble and form a lipid bilayer, and the proteins which are present in that, they act as enzymes and involved in the fast multiplication of molecules. Then what? Like this protobionts formed. Then what? Coaservatives. What do you call coaservatives? The nucleocoaservatives means pre-cells. These coaservatives plus nucleoproteins and nutrients are limiting. What do you call this point? You have to remember, limiting surface membrane all togetherly acts as a virus or free living genes. These all free living genes united to protoviruses present today present day so virus. What you call present day virus. These all points we should have to remember. Okay. So biological evolution. What do you call? Biological evolution. So next concept is, what do you call the next concept? What we are discussing is next concept, how the prokaryotes and eukaryotes are formed. This point we should have to remember. Okay, how prokaryotes and eukaryotes, these things you should have to remember. Two major cell types. Two major cell types. Okay, what are the two major cell types? That one we have to discuss. Okay, so one form of the how two major cell types, okay, means very well how prokaryotes and eukaryotes formed. That one we are going to discuss. Okay, one form of the nucleoproteins. First, very well nucleoproteins, as it is nucleoproteins. Okay, nucleoproteins embedded in the cell membrane. Embedded. This one you have to remember. Embedded in the cell. Embedded in the cell substance. This is the point you should have to remember. Okay, so next, what happened? Substance. So cells were similar to very well Monera. What happened? The Monera. Next, Next, modern bacteria. These are considered as which one? Modern bacteria. Modern bacteria. Then do you know very well blue-green algae? Blue-green algae. So viruses after these all the things happened. Next one, other form we are going to discuss. The other form, what is that one? The nucleoproteins. Nucleoproteins, as it is nucleoproteins. Okay, these nucleoproteins embedded from, okay, surrounding by a thin membrane. Surrounding by a thin membrane. So you know very well, surrounding by thin membrane means automatically it's a nucleus membrane. So then what happened? This one, and you know very well the protista. This one will be considered as a which one? Protista. Then later these two, automatically these two after what happened? Nucleo, what you called, natural sources declined. Natural sources declined. This point you should have to remember. Natural sources declined. Okay, then what happened? Ancestors of Monera, ancestors of Monera, very important, and Protista. Ancestors of Monera and Protista had to evolved. So evolved, okay, and they started what you call for evolved for what? Procurement of what you call the procurement of, this is the most important, procurement of food. Procurement for food, what they done, this is the important. So they changed into, or they converted into parasites, and they performed parasitism. Okay, and what he called saprophytism. Then what he called predator or amensalism. Okay, animalism, simply you can say animalism is short. Okay, and you know very well chemosynthesis, and or what you call photosynthesis also they performed. This is the most important. Okay, so the photosynthesis there, in the sense automatically what will happen? Oxygen will be increased. So in the oxygen increase in the sea atmosphere. This is the point you should have to remember. The sea atmosphere. So this one react with the CHO and O2, then convert into CO2 plus 2H2O. This is important. And 4NH3 react with the oxygen, okay, converted into 2N2 plus 6H2O. Like this, the CO2 formation, okay, oxygen combined with what you call oxygen combined with methane, carbon dioxide formation, and nitrogen formation we identified. So this one leads to large amount of energy. So then what happened? Large amount of energy. Okay, then what happened? Oxygen more means automatically aerobic respiration. Aerobic respiration means that leads to what? Production of more energy, large amount of energy by the oxidation of foodstuffs. So like this, what happened? Prokaryotes and what do you call eukaryotes evolved. Prokaryotes and eukaryotes evolved. These points you should have to remember. Okay, now come to the concept, please. So what happened? Preservatives, Okay, this is a probionts or preservatives formed. That one we discussed just one. Okay, protobionts. So first protobionts after how the viruses developed. You know very well viruses. Protobionts, coaservatives. Okay, coaservates. Then viruses. After that time, what happened? Some changes. So two major cells formed viruses to after two major cell types. How? The internal atmosphere forms nucleoproteins. They embedded in the cell substances without any membrane. So they will be the nuclear membrane just only that is there in that's it in the substance nucleoproteins. These are nucleoproteins embedded in the cell substances. So this is the membrane, but they are not covered with any membrane. So that's why they will be considered as which one? Munira. Munira without nucleus, nuclear membrane or without proper nucleus, just simply nucleoproteins like this. So now we are calling their modern bacteria or blue-green algae. Later, in some cases, next like this only, this is the what he called the cow cell. This cell surrounded by nucleoproteins also there. These nucleoproteins surrounded by what happened? The thin membrane. That time, those particular days, this thin membrane is there in the sense you know very well. That is nuclear membrane. That is called nuclear membrane, and what you call this is nucleus. So this particular one category comes under which one? Protista. So later natural sources declined. For them no food. So what in the sea, what happened? Natural sources, nutrient sources, natural sources are declined. So these ancestors of Monera and Protista, what happened? Evolved for procurement of food. To take the food from others, they showed some changes, modifications, some adaptations, some modifications, some adaptations. They changed like that. Evolved means some modifications, some changes happened. So they showed what kind of three actions? They showed what you call parasitism. You know very well parasitism. What is the parasitism? They will be live in the host. Okay, parasitism means they will be live in the host. Best example, you know, bacteria. What they will do? They will live in our body and they will gain the nutrients. They will change their body structure according to their internal body, our body conditions, and they will slowly take the food whatever we are taking, that one they will utilize and they will multiply and they will cause the disease like that. These all Monerian Protestants, for the procurement of food, they evolved and they showed the what kind of mechanism? Parasitism and dead bodies decomposition point of view, saprophytism, and predator, you know very well, prey and predator, they will kill the prey, and animalism, like they will eat the herbivores and carnivores like that. So these all the things happened. So next one, not only this, they showed the chemosynthesis and photosynthesis. What is the meaning of chemosynthesis? By utilizing chemicals, they produce the food, that is called chemosynthesis. By utilizing the light energy, here the chemicals, they are the light energy, then they produce it, they developed into new animals, other new what you call varieties. Okay, so like this photosynthetic bacteria, chemosynthetic bacteria developed. So automatically when the photosynthesis increased, when the photosynthesis process is there, what is the photosynthesis in the plants? What gas is releasing after photosynthesis? Oxygen. So then the oxygen increasing in the sea atmosphere because of photosynthetic bacteria, this free oxygen reacted with the methane, then formation of carbon dioxide, ammonia, then formation of nitrogen, like this kind of substances. And one more important thing is when the oxygen is more, they performed what kind of respiration? Oxygen is there in the sense what we call the respiration, that is called aerobic respiration. Then automatically the increase of energy leads to what metabolic activities, the energy utilized. Like this, what happened? The prokaryotes and eukaryotes evolved, where first protobionts to coaservatives to viruses, viruses to Monera protista. Then the ancestors of Monera protista evolved and converted into chemosynthesis, photosynthetic bacteria. Because of photosynthesis, oxygen increased. That oxygen reacted with the methane and ammonia, then forms the complete present day prokaryotes, and eukaryotes evolved. This process only called, what do you call this, is called biological evolution. Protobionts to prokaryotes and eukaryotes evolved. This is called biological evolution. The next concept what we are going to discuss is experimental approach. So how and who developed, who given the experimental evidence about the life, that is the most important. So this is the protobionts and conservatives, like that we discussed. We want experimental evidence for these all the things. How and what who done the experimental evidence, how these organomolecules are formed, how the DNA is formed, how the proteins are formed, that one we are going to discuss in this content. So first one, Urey and Miller. Is the scientist Urey and Miller. Experimental, what do you call it, approach, experimental approach to the, what you called, origin, to the origin of life, origin of life. Okay, now observe very carefully. So first, who are the scientists? Urey and Miller. Urey and Miller, these are the scientists. Okay, so they given the synthesis of organic compounds. How organic? Because the life formed from organic compounds only. How these organic compounds are formed. Okay, so that leads to the how the living organisms are formed from living organisms, from organic compounds. This one, organic compounds. That explanation given by who the Urey and Miller. Okay, see now what is the first one? Mixture of gases. What are the requirements for his experiment? What they did? Mixture of gases. They took number one, mixture of gases, and what he called the electric discharge. Electric discharge. How this they created electric discharge? The person Miller, tungsten electrode he used. Tungsten electrode he used. The next one, you know very well, very important. So small flask, he took the small flask. Then what he done, kept boiling. Okay, this is a very important. Water containing small flask, kept boiling. Next, and the steam. Then what will come? You know very well, steam. The steam will be come out. This steam, okay. Then what he called mix with the steam with the mix with the, you know very well, mixture of gases. These points you should have to remember. The mixture of gases. What kind of gases? Most important. This is the most important bit. This is the need bit. What are the mixture of gases are there? Ammonia. Okay. Then methane. Okay. Ammonia, methane, and what you call hydrogen. These three gases mixture they used. Okay. In the large chamber. Which one? Large chamber. This point also you should have to remember. Okay. Then the steam condensed. Then what happened? The steam condensed. Okay. Steam condensed. Next, to form water. Water formed. Then ran to the, which one? U tube will be there. One U-shaped tube also arranged in that experimental apparatus. So like this tube, then 1 week he kept like this. So then later that liquid analyzed. That liquid what he done? analyzed. So in that liquid, what he identified? Number one, formation of amino acids, you know very well, glycine, and what he called alanine. What he done? Glycine, alanine, and beta alanine. These are all the important, you should have to remember. Okay, beta alanine and aspartic acid. What he called? Aspartic acid were identified. So like this all considered as which one? Abiogenesis. Like this, abiogenesis will be happened by the help of which one? Large number of organic compounds. Organic compounds, like this organic compounds only given the life. So the next one, the source of the carbon. What is the source of carbon here? What is the source of carbon? You know, methane. Methane gas, source of carbon. Source of carbon is methane. Like that. So all types of amino acids they identified, observing that all type of amino acids and nitrogen bases. What he called, you know very well, nitrogen bases. What are the nitrogen bases? Purines, come on, purines and pyrimidines they identified. Purines and pyrimidines they identified or noticed. So this is the experimental approach. First what done? The Urey and Miller. Urey and Miller saying that identify what you called living materials formed from organic compounds. Living materials or life is coming from organic compounds, like that they said, and conformed with the experiments also. What he took as a operators are the requirements for their experiment is, in his operators, first they took the mixture of gases. What kind of gases? Mixture of gases. Come on, ammonia, methane, hydrogen. Then electric discharge. That time thunders are the electric power, but here he took same like that electric discharge. The tungsten electrode that he took in his, he fixed in his operators. Then he took the small flask with the water and kept boiling. Then automatically steam will be released. That steam mixed with the mixture of gases like ammonia, methane and hydrogen in the large chamber. Small flask water and the large chamber ammonia, methane and hydrogen. Then steam condensed, then water formed, then that water formed enter into the U-tube. 1 week identified that water and the mixture of gases all together mixed, and 1 week after he analyzed the liquid, that solution. That liquid analyzed. In that liquid, what the Urey and Miller identified? Formation of amino acids. What are the formation of amino acids? I will draw the picture and I will explain later, but this information you should have to remember. So glycine, alanine, beta alanine, like the aspartic acid, like that, you know very well, these are the most important amino acids. These amino acids togetherly combined, you know very well, what will form? Proteins formed. So like this, the theory was abiogenesis from organic compounds, explanation given by life formed from organic compounds with experimental evidence proved by the Stanley and Miller. So here what is the carbon source in this experiment? Main we have carbon. Carbon is abundant in the living things. The carbon source is methane. Source of carbon is carbon is methane. And later same like that, so many other amino acids, the nitrogen bases, you know, purines and pyrimidines like adenine and guanine, cytosine, thymine and uracil identified. Protein is there, purines, pyrimidines are there, then automatically life formed. Like this, they Urey and Miller did the experiment, and they told about these all the things. Okay, tomorrow I will draw the picture about this Urey and Miller and I will explain. Thank you children. Thank you very much. Next time we will meet.